And so with this, I'd really like to introduce our keynote speaker. It's my sincere pleasure to introduce Dritan Aqalliu. Dritan is familiar to many of us, first for his really excellent science and contributions to the field of brain barriers, and also as a past organizer of this meeting. He really did a terrific job co-organizing the last two meetings, and we were really fortunate with Benoit to have this training session with Dritan last time. So Dritan is originally from Albania, and he had obtained a degree from the University of Tara School of Medicine. And after that, Dritan moved to the US, where he carried out additional undergraduate training, bachelor degrees in genetics and development at the University of Minnesota. From there, he then continued his travels west, ended up at Stanford carrying out a PhD in the lab of Ben Barres. No, I'm sorry. We will get a correction on that. And then started his research lab at UC Irvine, and in 2015 he transferred from UC Irvine to Columbia to the Department of Pathology. And through his time, he's really focused on understanding the basic mechanisms, cellular and molecular mechanisms that regulate brain barrier formation, with a focus on Wnt beta-catenin signaling. And he's really interested in understanding the basic mechanisms of development and also the processes of barrier breakdown in a wide range of neurologic diseases, including multiple sclerosis, autoimmune encephalitis, and others. And his lab really takes us, they really leverage a variety of genetic, molecular, and cellular approaches to tackle these very challenging questions. And so he's currently associate professor in the Department of Neurology and Pathology and Cell Biology at the Columbia University. And we are really excited to welcome you back as our keynote speaker, and we're really excited to hear about the latest exciting developments from your lab. Welcome, Dritan.
Thank you so much. Goes forward, this goes back. Okay. Oh yeah, I can carry this one. So where's the first one? Oh. So first, thank you everyone. I would like to thank the organizing committee, Maria, Benoit, Julie, and Joey, for giving me this great opportunity to be the keynote speaker. I feel like, I'm sort of not at the level where I can give sort of this grandiose talk about, achievements, but I would like to, nevertheless, praise and some of our work that is happening in the laboratory. So I think, so what I'd like to do today is really give you a bird's eye view initially on some of the fundamental mechanisms of the blood-brain barrier, and really focus on trying to discuss some of the mechanisms that regulate the angiogenesis and the development of the barrier, as well as in the second half of the talk, I'm going to give you a story about mechanisms of blood-brain barrier dysfunction in autoimmune encephalitis. As we all know, and this is not new to this audience, obviously the central nervous system is a highly vascularized organ. Basically it has been estimated that the distance of a neuron from a cell is about 10 micrometer, and this exuberant vascular network is important because the central nervous system demands a lot of energy for propagation of action potentials and synaptic transmission, yet it cannot store energy. Astrocyte can store very small amounts of glycogen, but it requires constant supply of glucose and needs glucose. And so in order to address this, basically the neurons have to be in close proximity to the vasculature such that the process of neurovascular coupling happens. But in addition to the neurovascular coupling, Another feature that is unique to the CNS blood vessels is the presence of this barrier. So what you're looking here are two images. The images on the left is two-photon imaging of the cortex of a transgenic mouse that was generated in the lab a while ago that has a fusion of green fluorescent protein with one of the major tight junction-associated proteins called Claudin 5, and that allows us to visualize tight junctions of endothelial cells. And then in red, we have injected a tracer, small molecular weight tracer, biocytin tetrameterodomine, that allows us to do two things. One, to visualize the vascular tree, but then also to function as the blood-brain barrier. And as you can see in the healthy brain, this tracer is contained within the vessels, but in the context of a disease, in this case is an animal model for ischemic stroke, you can see basically that the tracer is leaking out. So barrier dysfunction is a key feature of some of neurological diseases. One of them is ischemic stroke. Another disease where there's a major dysfunction of the blood-brain barrier is multiple sclerosis. So here what you're seeing are two images basically of a healthy spinal cord in this case and an animal model for multiple sclerosis, experimental autoimmune encephalomyelitis. And you can see that basically at the peak of the disease there is a virtual complete disintegration of cell-cell junctions. So what I want to do today is cover these two particular topics. In the first half of the talk, I'm going to discuss some of the signaling pathways and mechanisms that regulate the angiogenesis and neurovascular barrier maturation. And in the second half, I'm going to talk about blood-brain barrier dysfunction and immune crosstalk with the CNS vasculature in the context of neuroinflammatory disease called post-infectious autoimmune encephalitis. As we know, the process of vascular development occurs in three specific stages. First is the stage of vascular growth and the selection of tip and stalk cells. The second phase is the process of remodeling, where the specification of the large arteries, veins, and capillaries is established, and there is the vascular pruning. And the third phase is the acquisition of the barrier properties, which organ-specific properties, which in the central nervous system, this relates to the acquisition of the barrier properties. So when we think about the barrier properties, there are four major characteristics that allow the endothelial cells in the central nervous system to have barrier properties. One is the presence of very tight junctions that are illustrated here that restrict paracellular diffusion from the blood into the brain. They also serve as fences to basically prevent the movement of the transmembrane proteins from the apical to the lateral side. The second is basically the presence of or the absence of caveolum-mediated transport that together then these create a barrier to bringing a lot of nutrients in the brain. And so to overcome this, the CNS endothelial cells express a variety of transporters that transport the nutrients from the blood into the CNS. A fourth property that isn't illustrated here is actually the absence of expression of a large number of cell adhesion molecules for immune cell adhesion and trafficking. And so we can visualize some of these through the transmission electron micrograph. You can see here a nice capillary surrounded by astrocyte and foot, and we can visualize here the tight junctions and very few caveola. Now, the acquisition of these properties, even though at a cell biological level, the barrier properties reside in the endothelial cells per se, I think the interaction of the endothelial cells with other cells of the neurovascular unit, pericytes and astrocytes, via the vascular basement membrane is essential for the acquisition and the maturation of the barrier properties. And so in the first part, I think I'm going to discuss some of the questions related to the development of the barrier and how this unit comes together to form a functional barrier. And to do that, I will primarily focus in the retina, because retina is a very nice system. It's part of the CNS, but it's a nice system because it has a very clear vascular tree. The development of the vascular tree in the retina begins postnatally, and the endothelial cells come from the optic nerve, and then during the initial phases of postnatal development up till P8, you can see the development of the superficial vascular plexus. And then later you form basically the intermediate and the deep plexi. So, of course, the development of the vasculature in the central nervous system doesn't happen in a vacuum. And so when we look at the time course of when this process of vascular development and neurovascular barrier maturation occurs in the retina, you can see that it's overlaid with the development and maturation of different CNS cell types, including neuronal cells, particularly bipolar and ROS. The other cells have already formed, and then the development and maturation of the glial cells. So work by a large number of people have already established that indeed endothelial cells cross-communicate with other cell types in the retina, and this is also true in the central nervous system. And this communication occurs via a large array of signaling pathways. And several of the signaling pathways, such as VEGF, is important for the process of angiogenesis. And the one that I'm going to focus today is the Wnt in beta-catenin signaling. In this case, I'm going to focus in the Norrin, because Norrin is the ligand that activates the pathway in the retina. So classically, this pathway requires the interaction of a ligand Wnt7A, 7B in the cortex and Norrin in the retina and cerebellum. And they require several receptors, Frizzled LARP. What I haven't depicted here are the GPR124 or EREC for simplicity. And basically, the interactions of the ligand with the receptor lead to translocation of the axin to the plasma membrane, and basically the dissociation of the beta-catenin destruction complex, which then stabilizes beta-catenin and translocates it into the nucleus. And so work by a large number of people, some of those are here in the audience, have already established that this pathway is essential for both CNS angiogenesis and blood-brain barrier development. And this pathway is highly active in CNS endothelial cells. But one of the questions that one of my postdocs in the lab had, Saptarshi Biswas, he's here in the audience and giving a talk tomorrow, is whether, in addition to endothelial cells, mural cells can also express or respond to the canonical Wnt signaling. They do express the receptors. They're exposed to the ligands, and so one of the questions that we had is to what extent mural cells that are adjacent to endothelial cells respond to this. So what I'm showing here is actually a staining for LEF1 and PDGFR-beta, which is a marker for mural cells. And what you can see here is robust LEF1 staining in endothelial cells, but also you can see LEF1 a little bit weaker in pericytes. And similar to what happens in endothelial cells, also in pericytes, you can see that as the retina develops, going from early postnatal P6 to P14, similar to the case with endothelial cells, the activity of the Wnt beta-catenin signaling goes down. So when we looked at the expression of the activation of the Wnt beta-catenin signaling along the vascular tree in arteries and capillaries, what was interesting and quite surprising was indeed the higher levels of expression of LEF1 in mural cells was in arteries and veins, and much smaller was in capillaries. And so we're basically sort of one of the initial questions that we had is that, the activation of the Wnt beta-catenin signaling goes down as the vascular tree matures and the acquisition of the barrier properties begins. And so how is this process really regulated? How do vessels know to grow but not mature barrier properties? And so the answer to this came from one molecule, APCD1, which my interest began when I was a postdoc with Ben Paris at Stanford University, and this was a collaboration with Richard Daneman because APCD1 was one of the top BBB genes that he identified in a screen for brain-specific transcripts. And so basically, to understand the function of APCD1 in this process, at the time we showed that indeed APCD1 is a regulator of the is a negative regulator of the canonical Wnt signaling. And so to understand its function, we first look at its expression. And in the retina, you can see that APCD1 transcript is expressed in the vasculature. When we look by a double fluorescent in situ with APCD1 and markers for the endothelial cells, pericytes, and astrocytes, it's very clear that APCD1 is expressed very highly in endothelial cells because it's a readout of the canonical Wnt signaling. It's expressed also in pericytes, but it's absent in astrocytes. So astrocytes don't respond to the canonical Wnt signaling. They also don't express APCD1. This is obviously during development. So in the mutant, what we saw, and I'm going to go fast because this has been published, basically that APCD1 mutants show a hypervascularization due to basically defects in basal pruning. Now the surprise came when we examined the blood-retina barrier properties of APCD1 mutants. So to do that, we injected the small tracer biocytin tetramethyrodomine, which I introduced at the beginning of the talk. And what we saw that indeed in the mutants, there was a reduced leakage of this tracer at P14 and P10 compared to the wild type, suggesting that indeed there was a decoupling between angiogenesis and blood-retina barrier maturation. And so basically, our studies then initially sort of concluded that indeed APCD1 is critical to put a brake on the canonical Wnt signaling such that the vessels that are high in Wnt are the vessels that are stabilized, they don't prune, and they form a barrier, whereas the vessels that have lower levels of the Wnt signaling are the ones that are going to be pruned and eventually eliminated. But the question that we had is, how does this happen at the molecular level? And so to perform these studies, we did bulk RNA sequencing between the wild type and the mutants, and then for each of the differentially expressed transcripts at different times of development, P10 and P14, we assign identity of this transcript based on the single-cell RNA-seq database of the retina. And so what was interesting is from P10, but also at P14, you can see that there are differentially expressed genes that are upregulated or downregulated in the mutant, both in endothelial cells and the mural cells. And some of the genes that are mostly differentially expressed are actually genes that are part of the extracellular matrix. So one of them called the attention of Saptarshi because this was LAMA2, which is a gene that encodes for one of the most prominent basement membrane proteins, laminin alpha 2. And so we used the transgenic mouse to basically visualize where LAMA2 is expressed. And what you can see here is when we stain for basically beta-gal, which labels, which is knocked into the LAMA2 locus, you can see that basically it colocalizes very nicely with pericytes. It's also expressed by some astrocytes, suggesting that indeed laminin alpha 2 is made both by pericytes and astrocytes. However, astrocytes don't respond to the canonical Wnt signaling, whereas pericytes do. And so consistent with this, the bulk RNA sequencing data, what we did see by fluorescent in situ hybridization, indeed that the transcript for laminin alpha-2 was upregulated in Ng2 positive resides in the APCD1 mutant. And at the protein level, we saw a larger deposition of laminin alpha-2 in the basement membrane. And what I'm showing here are the arteries because the phenotype was more prominent both in the arteries and in the veins. So what is the consequence of changes in this vascular basal membrane and for the assembly of the neurovascular unit? So one of the interesting things is when we looked at P14 for genes that were differentially expressed in astrocytes, what we did notice is that indeed in the mutant, astrocyte markers that are more characteristics of the mature astrocytes were upregulated compared to the wild type, suggesting that indeed there was an accelerated maturation of the astrocytes in the mutant. And particularly one of these proteins, aquaporin 4, which is at the end, it was highly expressed, and we confirmed that here when you look at the immunofluorescence for aquaporin 4, that it's higher in the vasculature. It's also expressed by the Microglia. And furthermore, Rishi confirmed this by transmission electron microscopy, when you can see that indeed in the APCD1 mutant there is a higher astrocyte end-feet coverage of the vasculature compared to the wild type. And so overall, this data suggests that in addition to the roles that we have known and accepted for the canonical Wnt signaling and regulation of tight junctions, the suppression of transcytosis, something to consider is that maybe at the neurovascular unit, Wnt signaling is doing a little bit more and playing a role in the regulation of the vascular basement membrane proteins. And so through a collaboration with Elisabetta Diana, basically we had shown before that in endothelial cells, the activation of the Wnt beta-catenin signaling induces expression of this transcript called FGFBP1, which is required for the deposition of collagen 4, not for the expression, but for the deposition of collagen 4 in the basement membrane. And the work by Rishi recently showed that indeed in pericytes, at least the activation of the canonical Wnt signaling is critical for the expression and secretion of laminin alpha-2, and a work that I don't have time to discuss today, but it's published. He demonstrated that this is required for interaction with integrin alpha-6, and if you block this, you don't have a proper maturation of astrocytes. And I think this is important because, as some of you may know, changes in the matrixome or in the composition of the vascular basal membrane are characteristic of cerebral small vessel disease, suggesting that perhaps aberrant Wnt beta-catenin signaling may underlie some of these disorders. Okay, so the next thing I'm going to cover, and I'm going to cover just a few snippets of this because Rishi is going to talk tomorrow in the morning session, is one of the questions that he had is, we know that a lot of signaling is required, communication between different cells is essential for the acquisition of the barrier properties. But to what extent neuronal activity plays a role? And what is already been established is that cholinergic activity is required for basically angiogenesis and acquisition of some of the barrier properties in the retina. However, glutamate is the largest neurotransmitter, and so one of the questions that Rishi had is, what is the role of glutamate in the regulation of angiogenesis and PRB function? So to address this, and he's going to talk more about this tomorrow, he used mice that are deficient for this vesicular glutamate transporter called VGLUT1. And in these mutants, basically, there is no release of glutamate. And what he saw is that in the VGLUT1 deficient mice, there is actually reduced angiogenesis and also increased blood-retina barrier leakage, suggesting that indeed the levels of extracellular glutamate are critical both for the angiogenesis and the acquisition of the blood-retina barrier properties. So what is functioning downstream of glutamate? What was interesting is when we looked at the expression of norrin, you can see that the VGLUT1 mutants that basically don't have glutamate, norrin mRNA is decreased. But in the GNET1 mutants, this is the opposite mutation where you have excessive glutamate in the extracellular space. Basically, there is an upregulation of norrin expression. Consistent with this, when we look at the LEF1 signaling, you can see an upregulation of LEF1 in GNET1 mutant and downregulation in VGLUT1 mutants. And this is also consistent with single-cell RNA sequencing that Rishi will discuss more in detail tomorrow. And so our current, so just the final experiment on this is what he did is he did a rescue experiment when he activated the canonical Wnt signaling in the VGLUT1 mutant using the 6-bio. And you can see that when you administer this drug, you can rescue both the angiogenesis and the barrier properties. I have to say, I've never seen such more profound rescue in a lot of experiments I've done in my life. But this is actually extremely dramatic. And so the model that I think that we have to consider is that in the CNS, we have to consider activity. And in fact, the glutamate plays an essential role not only as a neurotransmitter, but also in regulating the process of angiogenesis and barrier maturation, likely through regulation of the norrin beta-catenin activity. Okay, so in the second part now, I want to switch gears and talk to you a little bit about some of the mechanisms that induce neurovascular dysfunction in the context of post-infections, autoimmune encephalitis. So obviously, the question of how infectious agents can trigger neuropsychiatric disease is not a new one, although has emerged as an important one recently because of this pandemic that we've lived for the last 3 years. And it's been clear that indeed infection, even SARS-CoV-2, which doesn't actually infect the brain, can trigger both neurological and psychiatric sequelae. But one of the pathogens that we've been interested for a very long time is group A strep, because group A strep is actually notorious for triggering secondary sequelae. So it causes rheumatic fever in the heart, glomerulonephritis in the kidney, scarlet fever, and then in the brain it triggers a complex of disorders, symptom scoria and pain, as which I'll describe in a little bit. And what is thought is that indeed this is a misdirected immune response where basically the immune system that is activated to fight pathogen recognize is epitopes that are present in various organs and attacks them. So symptom scoria is the neurological sequelae of rheumatic fever. It's very common after severe group A strep infections and targets predominantly the basal ganglia. And it's work by several people have actually shown that in symptom scoria there are circulating antibodies to D2, the dopamine receptors, that when they target and bind the receptor, they trigger this uncontrollable movement disorders. PET imaging studies have also shown that indeed there is activation of microglia and neuroinflammation in the basal ganglia. And in 1998, Susan Swedo at the National Institute of Mental Health, while studying symptom scoria, identified a different syndrome in children that she termed pediatric autoimmune neuropsychiatric disorders associated with group A strep. This is also caused by repeated bouts with strep infections. There are antibodies to the basal ganglia that target predominantly the cholinergic neurons. It's sudden onset and then can be quite difficult and controversial to diagnose. So my lab has been interested for a while for this, initially driven by the interest of very desperate parents trying to figure out what's happening with their children. And some of the key questions that we've been interested for a while in the lab to understand about these disorders is how do antibodies reach the brain? what drives and sustains microglial inflammation, and then can we, if we develop tools to understand the neuroimmune responses, can we then develop treatments or diagnostic tools? This disease really needs them. So to do that in the lab, when I started my lab at UC Irvine, we decided to tackle this question by developing a new mouse model for the disease. And in this model, we basically do intranasal infections with group A strep repeatedly for 4 weeks, because children get exposed to group A strep infections continuously. And that what this has shown is that indeed this is the nose. So the group A strep is only present in the nose. It doesn't have an affinity for the brain. It's not like group B, which you'll probably hear as well in this meeting. But basically in the nose, it resides in this organ called the nose-associated lymphoid tissue, which is the equivalent of the tonsils in humans. And the repeated infections are required to generate a proper Th17 immune response, because Th17 cells are required for fighting the pathogen. Now, given that Th17 cells play a critical role in a large number of other diseases, such as MS, we wondered whether indeed these Th17 cells can enter the brain, and then they can be the cause for brain barrier dysfunction. And a while ago, we actually, a very talented postdoc in the lab, Eric Smith, showed very nicely that repeated but not single infections with group A strep are sufficient to drive the entry of the T cells through the olfactory route into the central nervous system. And the presence of T cells in the brain is associated with a series of pathology. One is blood-brain barrier disruption. Second is microglial activation, and third is loss of excitatory synapses, which results in anosmia in mice. And so in order to understand more collectively at the single cell level, how CNS cell types are responding to this, a former talented graduate student, now she's a postdoc at Rockefeller University, decided to take a single-cell RNA-seq approach. And we did this both through all the cells in the olfactory bulb, but we also enriched for both endothelial cells and microglia, because those cells are basically relatively small in proportions. And so what you're seeing here is a t-SNP plot of different cell types, assigned through the assigned identity and separated by basically healthy or strep infections. And what I want to point out is the two largest populations that have the largest shift in differentially expressed genes are the microglial and the endothelial cells. So what's happening in this population? So when we look at the endothelial cell subpopulation, we can see that there are several Go terms that are upregulated related to the endothelial response to LPS, the antigen presentation, and then what are downregulated are terms related to the blood-brain barrier function and extracellular matrix. And so we confirmed this by fluorescent in situ hybridization. What I'm showing here are the Weiling plots for 3 BBB transcript, MFSD2A, which regulates transcytosis. That's the work of Cheng Guogu, and then the two most enriched BBB transcripts that we know, ITM2A and ITIH5. And both of them are, down in after group-based strep infections. So then what we did is we basically had performed in parallel work by Sanjay Sharyar. He is here in the audience. He did single-cell RNA sequencing of endothelial cells in EAE at acute and chronic. And so we did a correlation analysis to see what genes are, among the differentially expressed genes in the two disease states are they similar or are they different? And what it turns out is that indeed they are very similar, suggesting that there is a core neuroinflammatory response in endothelial cells that occurs regardless whether indeed this is due to secondary sequelae from an infection or neuroinflammation. There are some unique genes that are particularly upregulated in one versus the other condition. One of them is LCN2, which is basically highly upregulated after group A strep infections. So the second population that we looked in detail was the microglia, obviously, because of these reported cases in pediatric cases of increased microglial activation. So what you're seeing here is a t-SNP plot of the microglia, and you can see that there's a very clear separation of different populations. And when we look at the upregulated genes, they fall into these three major classes: the response to type 1 interferon, antigen presentation, and cytokine production. And in fact, we can distinguish 6 different subpopulations of microglia. 2 that are homeostatic. These are microglia that express the homeostatic, classical homeostatic genes, and they are present mostly in the healthy, and then 4 disease-associated, which we're calling them streptococcal-related populations. And as you can see here, basically most of the homeostatic genes are expressed very highly in the HMG1 and 2, which are the resting microglia, and then the disease-associated are in the upper 4. Similarly, when we look at the antigen presentation and type 1 interferon, those are also highly upregulated in the disease-associated microglia. So just to summarize this bit, I think the single-cell RNA sequencing is sort of confirming some of our previous findings that were kind of rudimentary, that indeed there are a dramatic shift in blood-brain barrier function after repeated group A strep infections, and that there is a pro-inflammatory response in microglia. So our previous work had shown that indeed this pathology is related to the presence of Th17 lymphocytes in the brain. And work by Marian Platt, a former grad student in the lab, showed that when you ablate Th17 cells using RR gamma T deficient mice, which is a transcription factor required for the specification of Th17 fate, you actually can rescue all of this pathology. And so we confirmed this at the single cell level, where you can see that in the RR gamma deficient mice, you have an upregulation in the homeostatic genes, a reduction in the disease-associated chemokine, cytokine in response to interferon. However, antigen presentation was off the roof, suggesting that somehow in the RR gamma deficient mice, microglia are highly expressing antigen presentation genes. So this was somewhat of a surprise. Now, given that Th17 secrete a variety of cytokines that are characteristics, we wondered whether indeed a blockade of IL-17, one of the major cytokines that is made by Th17 lymphocyte, would mirror the phenotype that we saw with RR gamma deficient mice. And so to do this experiment, Charles had basically administered a neutralizing monoclonal antibody to IL-17 twice weekly, starting before infections and at this pattern during the weekly infections. And what we did see is that indeed after, even though we were administering an IL-17 blocking antibody, we could still generate Th17 cells, and they entered the brain just fine, suggesting that indeed the migration of these cells into the brain is not related to the IL-17. But what was interesting to see is that indeed when we looked at blood-brain barrier function in the IL-17 administered, basically we saw a rescue of the BB leakage. And when we look at the microglial response, it's interesting to see that this also mirrors a lot of what happened with the RR gamma deficient mice, that there was an upregulation in the homeostatic genes and then a downregulation in disease-associated chemokines and cytokines response to interferon. But similar to the RR gamma T deficient mice, there was a high upregulation in antigen presentation. So what that suggested is that indeed IL-17, and in data that I don't have time to discuss today, but we have put this preprint in the bioarchive, so for those of you who are interested, you can look at the paper in a lot more detail. we did these experiments in the GM-CSF knockout mice where we eliminated GM-CSF in Th17 lymphocytes, and we didn't see the same phenotypes as IL-17, suggesting that among the Th17 secreting cytokines, IL-17 is the predominant relevant for the disease. So the question then comes, so what? How is this related or relevant to the disease? So for this, we collaborated with Susan Sweeter at the National Institute of Mental Health to basically, who provided us with sera collected at the acute phase of the disease in children who suffer from PANDAS. And in addition, we also collected, the sera from children who came to the pediatric neurology clinic at Columbia. And what was interesting is that and I'm showing just here a subset, what was interesting is several of the cytokines that are also upregulated by microglia and macrophages were highly elevated in sera from patients, including CCL2, 3, 4, and 5, CXCL10, and then obviously some of the classical T cell ones, IL-6 and GM-CSF and IL-7. And so the question is, can these be used as biomarkers for the disease? And that's something that we want to see in the future. But one of the things that we want to test is also, what effect do the sera from the children have on the BBB properties? And this has been an eluding question for us for a while, because we wanted to develop a model of the human BBB that mirrors what happens in vivo situation. However, it was very challenging. And it was challenging because basically through a study that we did in collaboration with Raphael and Andrew Sproul, Raphael is here and Tyler will be here, we found that indeed a lot of the protocols that were available to the community generated either barrier-forming epithelial cells or endothelial cells that lack brain organotypicity. And through a consortium collaboration funded by the NHLBI that has Columbia, myself and Dr. Andrew Sproul, Dr. Erin Peterson at Scintillon, and Raphael at Cornell, our goal is to really kind of develop protocols where we can basically force brain organotypic traits out of generic endothelial cells. And the approach we're using is twofold. One of them is basically developing this protocol, and this is at Columbia. You're going to hear a lot more about this in session number 2, poster 101 from Ronak, who is here in the audience. But basically the protocol is to generate generic endothelial cells using this well-established protocol and then force them to acquire BBB properties by basically transducing them with BBB-specific transcription factors FOXF2 and ZIG3 and exposing them to a Wnt agonist. And we are getting some of these properties. So what I'm showing here is basically these are the generic endothelial cells, and then these are the ones that express BBB properties. And you can see that some of the transcripts that are highly expressed in the BBB begin to express. And these cells also acquire a higher transendothelial electric resistance. The second approach that we've taken, and this is in collaboration with Cornell, Raphael and the Scintillon Institute, and there are 2 posters in this session about this, is to basically take the protocol that generates these epithelial-like cells and then force them to become endothelials through expression of the 2 EC-specific transcription factors, SOX7 and ERG. And then interesting is once you generate these generic endothelial cells, if you co-culture them with pericytes and astrocytes, we find that this co-culture is sufficient to drive some of the BBB characteristics in these cells. And so with these cells in hand, what we're doing right now is that we're using basically the Mimetas platform, which has been very successful in our hands, to build this 3D neurovascular unit. This image is a courtesy of my colleague Irene Pedersen from the Scintillon Institute, who is following us online. But we can nicely get like tubules of endothelial cells that express tight junctions, and they interact very well with both pericytes and astrocytes. We're not perfect, but we're trying to get there. And so we have used this then to ask them the question, how do the sera from children who suffer from PANS or PANS affect the barrier properties? And so to do that, we can inject tracers in the tubule, the blood size, and measure the diffusion of these tracers across in the intermediate and in the bottom channel. And what's interesting to see is when we look at this diffusion of this tracer 70 kilodalton extran, you can see that this tracer it diffuses at a much higher rate when the 3D system is infused with sera from PANS children, suggesting that indeed at the acute phase of the disease, basically the expression of the cytokines, that is characteristics of microglia or macrophages, may drive in fact barrier dysfunction and may basically promote antibodies to enter the brain. So with that, I want to highlight some of the people who have done the work, going through many generations. This is the current lab, and these were the former labs. But basically, I want to highlight, particularly for the development, the work of Saptarshi Biswas, former postdoc, now associate research scientist. He is a fantastic scientist, and you'll hear more about his work tomorrow. He basically did all the studies on the role of Wnt signaling in mural cells and also on the activity. And some of that initial work was started by Jenna Mazzoni in collaboration with Sanjit Sharyar, who is here in the audience. A lot of the work on the post-infectious encephalitis was started initially by a very talented graduate student, Marianne Platt, who is now a postdoc at Yale, and by a fantastic graduate student, Charlotte Wayne, who is just spectacular, and she's now a postdoc at Rockefeller. And a lot of the work that we're currently doing is a collaboration of different people. This was a collaboration with Andrew Sproul and Ron Atel, Raphael Lisa at Cornell, Erin Peterson, and Scintillon. And then I want to highlight the continuous collaboration with the computational biologist, Vilas Menon. And some of the work I didn't describe, we also did Murphy's analysis in collaboration with Doris Schaefer to look at spatially where these microglia activated transcripts are regulated. And if you're interested, you can look at that publication. I want to highlight some of the team, Amang, who is here, and Ugur, who unfortunately, he was planning to come here but couldn't come. These have sort of pioneered a lot of the work we're doing on the METAS. And then I also want to highlight Mary Claire, a grad student who is working on stroke and type 1 interferon. So if you're interested, please visit her postdoc as well. Obviously, this could have not been done without funding, which is highlighted here in red. And I also want to be thankful to the parents who, despite the the adversity and all the suffering, they do a lot of fundraising to bring funds to the lab for this research, which unfortunately is not well recognized by the NIH. Thank you.
All right, thank you, Dritan. That was a wonderful talk. Great to set us up with so many different techniques. So we can go ahead and take questions from the audience. Brandon. Oh, you need a microphone. I'm faster. It's a race. I got to race the guys.
Hi, Dritan. Really great talk.
Thank you.
I just had two quick questions for you. One of them was, do you think that the downregulation of MFSD2A that you saw with group A strep infection is the potential mechanism of the autoantibodies transiting the blood-brain barrier?
Potentially. It's a little bit more complex than that, but one of them for sure. There are other changes that occur. Definitely there are changes in some of the tight junction transcript, changes in several of the basement membrane proteins. So there are several mechanisms that are at play, but definitely one of them.
Cool. And then second quick question, you showed that the PANDAS patient serum was sufficient to initiate leakage in your improved model. Were the cytokines themselves sufficient? Like, did you just do the cytokines, or was there something else in the serum that might be contributing?
No, we did. So the cytokines, we didn't do it in the 3D model, but we did it with a monolayer, and cytokines can upregulate transcytosis very, very robustly, actually. We use them at the concentration that we could detect them in the patients here, and they're very robust at doing that. Particularly CCL5 is just madly triggering this.
Cool, thank you.
Yep. But importantly, IL-17 in our hands doesn't do anything in vitro. Nothing. It doesn't trigger any disruption of the barrier. In fact, I have it here, since we are talking about that. Sorry, I'm going to skip that. Oh, sorry. I'm going to go here. Yeah, so you can see basically this is IL-17. IL-17 does absolutely nothing. It doesn't change the transendothelial electrical resistance. It doesn't change the transcytosis. So we don't think IL-17 is doing this directly. It's doing it indirectly through activation of microglia. Yeah?
My question is about your APCDD1 inhibition of aquaporin-4 and the loss of end feet.
Mm-hmm.
We have aquaporin-4 knockout mice, and yet the end feet are intact.
Mm-hmm.
And so how do you reconcile these?
So it's not that we see a loss of end feet, right? But it's not just aquaporin-4. There's a lot of other proteins at the end feet complex that are changing in this mutant. So it's not just aquaporin-4. I brought aquaporin-4 to highlight one, but integrins are changing, okay, and other end feet associated proteins are changing as well.
Okay, thank you.
Here. Beautiful talk here. So,
yes, sorry.
So BBB dysfunction happen to many dimensions, such as Alzheimer's disease. So in this type of context, how we look at your Wnt signaling in terms of like APCDD1 expression?
I haven't, but I'm sure you'll hear talks on it from this audience, so I'm sure you'll hear.
Thank you.
Dritan? Okay. Hi. Thanks. So I'm curious, you showed astrocyte contribution in the retina, but there's a profound difference in timing, and correct me if I'm wrong. In the retina, the vasculature grows on a carpet of astrocytes. Mm-hmm. They're already there.
Yeah.
In the cortex, the astrocyte will be born much later than the vasculature is formed. Do you have any idea of what this difference would be to the signaling pathway?
In the cortex,
yes.
You mean? So a lot of things are preserved in the cortex and cerebellum. For the sake of time, I didn't discuss them. You can check the paper. But basically what we did find is the mural cells also activate when signaling in the cortex and cerebellum, and changes in aquaporin-4 are very prominent in the cerebellum as well. So it's not just retina specific.
I'm asking about the timing. So the specification in the cortex, the pruning, and everything else will be happening later, in the absence of astrocyte altogether.
Will it? Everything?
I don't know. Well, to a certain time point, right?
I mean, you know, some of the barrier properties do occur in the presence of astrocytes as well.
Okay, thanks.
But fundamentally, there is a major difference. You're right in that astrocytes are there in the retina and they're not there in the developing cortex for sure. So basically, some of the more mature BBB properties, like transporters, would occur when the astrocytes are there. Yeah? Oh, sorry.
Thanks. That was a really interesting talk. And I was wondering, in past meetings, we've had discussions about the challenges with having and generating the in vitro BBB models that faithfully recapitulate endothelial cells and the whole structure. And I was wondering if you could comment on the developments you've made in your preparation to help overcome some of those challenges?
So I will not, because I want people to go to the posters. And so I wanna let basically people to like be excited about the posters. But generally speaking, the two approaches we took are, given that we know from the BBB transcriptome, we know a set of transcription factors that are uniquely in the brain and that genetically are important for this, such as FOXF2, SIG3. So that's basically, we figured that maybe we can force, by transcription factor reprogramming the generic endothelial cells to acquire some of the BBB properties. And they do acquire, but not everything. So I think maybe we haven't been clever enough to figure out the exact repertoire that is needed to induce this, or that we're just doing the sequencing with astrocytes and pericytes as well. But at least from the other model, which basically we took the protocol that generates these like epithelial-like cells, and then we force them to become endothelials by SOC7 and ERG. And even those generic endothelial cells tend to be very plastic, so in the presence of astrocytes and pericytes, they can acquire some BBB properties. But I think at the end, there might be a combination of a few different approaches that we take. We're still figuring out the best way, and there might not be a best way there, So, we might approximate something, but we might not be able to achieve what we want to achieve, which is to get as closer to the brain as we can. But I think that's what we're going to try.
Thanks. We'll go to the posters too.
Please do, actually, because I didn't even tell you a tenth of what you'll see in the poster. I just wanted to give you snippets so that, but it also will stimulate discussion for the other sessions, in particular also using these models to understand drug transport across the CNS as well.
All right, well, let's please join me in thanking Dritan again for a fantastic keynote talk.